Welcome back to the deep dive. Today, we're tackling maybe one of the biggest headaches in modern biology, trying to make sense of all this data. Specifically, how do we link what a cell is genetically with where it actually lives in a tissue?
Exactly. It's a huge challenge. You've got these two amazing technologies, right? First, there's single-cell RNA sequencing, scRNA-seq, gives you incredible detail about individual cells, all their differences.
Tells you who the cell is.
Right. But the process itself, it rips the cell out of its home. You lose all the spatial context. The map is just gone.
And then, on the flip side, you've got spatial transcriptomics, ST. Now, that gives you the map. You know where the gene expression is happening in the tissue slice.
But the gene information itself can be, well, let's just say, less precise, often noisy.
So, we know who but not where, or where but not really who in high fidelity. The core problem is finding a way to represent a cell mathematically that captures both its detailed gene profile and its real relationship with its neighbors, the whole tissue context.
And that's where this new framework our sources describe comes in. It's called TopoLa. Stands for Topology-encoded Latent Hyperbolic Geometry.
Okay, TopoLa. It sounds like it's fundamentally changing how we measure how similar cells are. So our mission for this deep dive is to really unpack TopoLa, figure out how it captures these fine-grained cell-to-cell topological relationships.
And then show the proof, how it boosts performance across actually 7 key biological analysis tasks. We'll see some pretty impressive numbers.
Let's dive in. The name itself mentions geometry. Latent hyperbolic geometry. That sounds complicated.
It is a bit different, yeah. So imagine looking at a tissue that's not just a bag of cells, it's a network. Cells talking to each other, influencing each other. TopoLa takes this complex cell network and maps it into a special mathematical space, a latent hyperbolic space.
Oh, hold on. Hyperbolic space, like curved space. Why not just use regular flat, Euclidean geometry, the kind we learned in school? That's what pretty much everything else uses.
It's a great question. Think about it like this. Try drawing a really big, complicated family tree, one going back centuries, on a standard sheet of paper.
Okay.
As you add more generations, more branches, things get incredibly cramped, right? You end up having to squash siblings together, stretch connections, just to make it fit on that flat surface.
Yeah, it becomes distorted, messy.
That distortion. That's basically the bias that traditional flat space models introduce when dealing with complex networks. Biological networks like family trees or signaling pathways between cells, they're naturally hierarchical. They have clusters.
Things group together, influence spreads out in branches.
Exactly. Hyperbolic geometry, because it's negatively curved, inherently has more room to accommodate these branching hierarchical structures without that forced distortion. So TopoLa uses this geometry to model the cell interaction more faithfully, correcting biases you get from just looking at expression data or simple distance.
Okay, that makes sense. Using a curved space fits the curved branching nature of biological networks. So if we're in this curved space, we need a new way to measure distance, a new ruler. How does TopoLa measure the distance between cells? It can't just be how far apart they are physically, right?
Correct. It needs to capture that network structure. And that brings us to TopoLa's first key piece. The TopoLa distance, or TLD. TLD is the metric for measuring distance in this latent hyperbolic space.
And how is it calculated?
Well, the math involves combining two main things. First, there's an energy term, which relates to things like spatial distance or maybe differences in gene expression profiles, standard stuff.
Okay, and the second thing?
The second factor is called the chemical potential.
Chemical potential. That sounds like it's straight out of physics or chemistry. What does that mean in terms of cells in a tissue?
Think of it this way. The chemical potential here is basically a measure of the cell's degree in the network, how many connections it has, how many neighbors it's interacting with.
So it's popularity or connectivity within the network.
Precisely. And this is really the clever part. TLD says the distance or similarity between two cells shouldn't just be about how different their genes look or how far apart they sit. It should also factor in how central or connected they are in their local network neighborhoods.
So two cells could be physically far apart, maybe even have slightly different gene profiles, but if they both play similar, highly connected roles in their respective parts of the tissue network.
TLD would see them as being topologically closer, more similar than a simple Euclidean distance would suggest. It's capturing a deeper relational similarity.
Right, that's intuitive. It's about functional or structural roles, not just raw distance. Okay, so TopoLa calculates this smarter distance, TLD. How does it then use TLD to actually improve the cell representations, to clean them up?
That leads to the second key component, TopoConv. It stands for TopoLa convolution. Essentially, the whole network structure, viewed through the lens of TLD, gets encoded. TopoConv then uses this unique TLD distance to perform a kind of spatial averaging or convolution.
So it's like it looks at a cell, and then it looks at its neighbors, but it weights the influence of those neighbors based on the TLD distance. Neighbors that TLD says are topologically important get more say.
Exactly. It aggregates information from neighbors, but it prioritizes input from cells that are structurally similar according to TLD, even if they aren't the absolute closest physically. It's refining the cell's representation by incorporating the sophisticated network geometry.
And you mentioned something crucial earlier, this enhancement, this TopoConv step. It doesn't actually change the size or dimension of the cell's mathematical representation.
Correct. That's a huge practical advantage. It means you can take TopoLa, calculate TLD, apply TopoConv, and use it as a pre-processing step for almost any existing downstream analysis pipeline or machine learning model. It just slots right in.
It's like a universal booster pack for cell representations.
You can think of it that way, yes.
Okay, let's just pause for a second and recap the core ideas, because this is foundational. We've got moving from flat Euclidean space to curved hyperbolic space to better represent network hierarchies. A new distance metric, TLD, that measures similarity based not just on proximity or features, but also on network connectivity or degree. A convolution step, TopoConv, that uses TLD to refine cell representations by smartly incorporating neighborhood information.
That's the essence of the framework.
Great. Now let's dig a bit deeper into the mechanism.
Okay.
You said TLD captures network structure. What specifically makes it better at this? You mentioned paths.
Right. This gets to the heart of why TLD is so effective. When it calculates the similarity or distance, TLD places a particularly strong emphasis on even hop paths in the network.
Even hop paths.
Yeah.
So connections that take 2 steps, 4 steps, 6 steps, as opposed to direct one-step connections. Why are those better?
Think about a social network again. If you want to know if two people really belong to the same community or share genuine interests, you don't just check if they're friends, a one-hop path. That could be random. You'd look at how many friends they have in common, right? That's a 2-hop path. Person A, mutual friend D, person B. Those shared connections, those 2-hop, 4-hop, even hop paths, they tell you much more about shared structure, shared community.
Ah, I see. It's about shared neighborhood structure, not just direct links.
Precisely. TLD prioritizes these even hop paths because they implicitly capture both the broader connectivity patterns and the degree, the importance of the cells involved. Cells with similar patterns of these even hop connections tend to have similar topological roles and end up closer together in the latent hyperbolic space.
It's like a mathematical fingerprint of a cell's place in the network's community structure.
Right.
Okay, that makes sense for TLD. Now, what about TopoConv? How does it actually make the representations mathematically better, more robust against noise? You mentioned something about singular values.
Yes, the singular value gap. This is a concept from linear algebra, from how we can decompose data matrices. Think of the matrix representing your cell network data. TopoConv, when applied, mathematically manipulates the singular values of this matrix. Specifically, it increases the gap between the important singular values, representing the main signal, and the less important ones, representing noise.
Can you give me an analogy for that? Increasing the singular value gap. What does that look like?
Imagine your data is like a slightly blurry photograph. The principal singular values correspond to the main subject, the core biological information you actually care about. The smaller singular values, that's like the background fuzz, the graininess, the technical noise, maybe batch effects.
Okay.
Increasing the singular value gap is like digitally sharpening the photo. It makes the main subject stand out much more clearly by relatively suppressing or pushing down the contribution of all that background noise and fuzz.
So it's effectively reweighting the information. Emphasizing the signal, de-emphasizing the noise, based on the network structure revealed by TLD.
Exactly. It's an intelligent, structure-aware noise filter. And critically, this isn't just a gut feeling or a heuristic. There's a mathematical proof behind it.
A guarantee.
Yes. By increasing this gap, the TopoLa-enhanced data representation becomes provably more robust against random noise, errors, even missing data points compared to the original input data. The math shows that the upper limit on how much noise can distort the distances in the TopoLa network is less than or equal to the limit for the original network.
So you are guaranteed to get a cleaner, more stable representation that's less susceptible to the inevitable errors in biological measurements.
That's the theoretical underpinning. Better geometry, better distance, mathematically proven noise reduction.
Fantastic. That sets the stage perfectly. We have this robust geometry-enhanced representation. Now let's see the payoff. Let's walk through these 7 biological tasks where TopoLa was applied and see the real-world impact. The big message seems to be its universality.
Absolutely. Let's start with task 1. Basic scRNA-seq clustering. The goal here is just identifying cell types from single cell data. They took 2 existing state-of-the-art methods, SIMLR and scGNN, and just added TopoLa as a pre-processing step.
And the results.
Pretty significant bumps. SIMLR improved by 5.8% in adjusted RAND index, that's a common clustering quality score, and scGNN improved by 6.1% in normalized neutral information, another key metric.
So just by pre-processing with TopoLa, these established methods got noticeably better at finding the right cell groups. Any specific examples?
Yes, there was a compelling case using the Purim dataset. It involves non-malignant cells in head and neck cancer. Originally, the standard methods tended to incorrectly mix T cells together with macrophages and fibroblasts in the clusters.
Which isn't biologically right.
No. But after applying TopoLa enhancement, those T cells formed their own distinct, separate clusters. They became spatially discrete populations in the representation space. It shows TopoLa's distance metric was successfully separating cells based on their true underlying network relationships, fixing that initial mix-up.
Cleaning up the classification purely through better geometry. Okay, what about bigger challenges, like dealing with data from different experiments or different types of measurements? Tasks 2 and 3, multi-batch and multi-omic integration.
Right, these are critical. Multi-batch integration is about removing technical artifacts, batch effects, when you combine data from different labs or experimental runs. Here, they enhanced a model called scGPT.
And the improvement.
An average of 15.4% better ARI across datasets. A specific example was the Cortex dataset. The original model wrongly split astrocytes, a type of brain cell, into 2 separate groups because of batch differences.
Making it look like there were 2 astrocyte subtypes when there weren't.
Exactly. TopoLa fixed that geometric distortion. With scGPT plus TopoLa, the astrocytes clustered together tightly, correctly, as one population. It effectively removed the technical noise while preserving the biological signal.
Okay, 15% is solid. Now, multi-omic integration.
Yeah.
This sounds even harder, combining, say, RNA data with protein data from the same cells. Totally different kinds of measurements.
This is where things got really dramatic. Using the BMMC dataset, that's bone marrow mononuclear cells, they combined RNA and protein, ADT data. The improvement with scGPT plus TopoLa was, well, it was a 45.7% increase in ARI.
Wow, nearly 50%. That's huge. Why is such a massive jump there?
Because when you have data from fundamentally different omics layers, finding a common ground, a shared representation space without bias is incredibly difficult for standard methods. TopoLa's ability to map both beta types into that consistent, latent hyperbolic geometry, guided by the inherent network structure, seems to preserve the true biological similarities much, much better.
So it found the shared structure despite the different data types.
It really clarified things. For example, distinguishing between CD4 plus T-naive and CD8 plus T-naive cells became much clearer. An improvement of that magnitude, nearly 50%, is, well, it suggests a major step forward for integrating diverse biological data.
Okay, let's shift gears slightly. Task 4. Rare cell identification. Finding those needles in the haystack, often the most clinically relevant cells, but so hard to spot.
Definitely high stakes. Here they looked at a method called surprisal component analysis, or SCA. SCA is designed to find cells with unusual or surprising gene expression patterns, which often highlights rare types.
But the risk is it just finds noise, right? Outliers.
That's the danger. SCA can amplify noise. But when combined with TopoLa SCA plus TopoLa, the F1 score, which balances finding the rare cells and avoiding false positives, improved by 28.1%.
So a big improvement in accuracy.
And crucially, it was about reducing false positives. In the airway dataset, for example, standard SCA flagged a lot of cells incorrectly. Adding TopoLa significantly cleaned that up, allowing for a much more precise isolation of genuinely rare cells like goblet cells or ionocytes. It filtered the noise, letting the true rare signal emerge.
Makes sense. The noise reduction aspect we talked about earlier really pays off here. Okay, finally, let's circle back to where we started, the spatial aspect. Tasks 5, 6, and 7 all focused on spatial transcriptomics data, particularly using those structured brain slices, DLPFC.
Right. So task 5, spatially informed clustering. Trying to identify distinct tissue layers or regions directly from ST data, which can be noisy. They enhanced a model called GraphST.
What was the key challenge here?
The most realistic scenario is when you don't know beforehand how many distinct layers or cell types there are. In that challenging setting, GraphST plus TopoLa showed a 20.9% average improvement in ARI.
So it was better at discovering the correct number and identity of spatial domains.
Yes, it became much more accurate. For instance, in those DLPFC brain slices, it could properly distinguish between layer 3 and layer 4, which can look quite similar in noisy ST data.
Okay, now what about building a 3D picture? Stacking multiple 2D slices that requires task 6, vertical integration, dealing with batch effects between slices.
This is another place where TopoLa really excelled. When integrating serial tissue slices, again using GraphST plus TopoLa, the improvement in the ASW score average silhouette width, which measures both batch mixing and biological structure preservation, was a massive 59.8%.
Whoa, nearly 60%. That's enormous.
It is. It means TopoLa was incredibly effective at harmonizing the technical differences between the slices while keeping the biological layer structures perfectly aligned, essential for any kind of 3D tissue reconstruction.
Absolutely critical. And the final piece of the puzzle, task 7, integrative analysis, combining the high detail of scRNA-seq with the spatial map from ST, using a model called IRIS.
Here, IRIS plus TopoLa achieved an 11.7% average ARI improvement. Again, looking at the DLPFC slices, the original IRIS model sometimes struggled, letting functionally distinct layers like layer 5 and layer 6 kind of bleed into each other in the integrated analysis.
But TopoLa helped.
Yes, the TopoLa enhanced representations allowed IRIS to achieve a much cleaner, more accurate separation of those layers, respecting both the gene expression detail and the spatial boundaries.
So across the board, basic clustering, integrating diverse data sets, finding rare cells, analyzing spatial data, combining single-cell and spatial TopoLa consistently provides a significant boost. The evidence seems really strong.
It really does. It paints a picture of TopoLa not just as a niche tool, but as a foundational improvement in how we represent cellular data by properly incorporating network geometry.
And the key takeaway you mentioned earlier, this isn't just for biologists.
That's what's potentially so exciting. The core methodology mapping a network to latent hyperbolic space, using TLD to measure distance based on topology, using TopoConv to enhance representations by boosting the singular value gap, that's fundamentally a general network geometry framework.
So the math isn't specific to cells.
Not at all. We talked about cell networks, but it could be applied to any system you can represent as a network or a matrix where understanding the underlying structure and reducing noise is important.
Which opens up, well, a lot of possibilities. If this geometry of relationships clarifies cell types, where else could it work?
You could think about core machine learning tasks, maybe improving link prediction in networks, predicting future connections, or enhancing the quality of word embeddings or graph embeddings used in natural language processing and AI, where capturing complex relationships between entities is key.
Or even things like analyzing social networks, understanding influence, community structure.
Or cybersecurity. Imagine using this to better distinguish real threat patterns from background network noise or deliberate obfuscation. The mathematical guarantee of robustness against noise is very appealing there.
It feels like TopoLa offers a new theoretical lens for looking at complex systems, suggesting that our standard flat space ways of measuring distance or similarity might be missing a lot of the real structure.
I think that's right. It really underscores the idea that to truly understand complex, interconnected systems, we have to consider the inherent geometry of the relationships themselves. That geometry holds hidden information.
Which brings us to our final thought for you, our listeners. If TopoLa reveals hidden biological structure by using a smarter, geometry-aware measure of distance and filtering noise, think about your own world. Where might you have complex data, networks, or relationships maybe in your work, in social structures, supply chains, anything where looking beyond simple similarities using a lens that captures the underlying geometry of connections might reveal a deeper structure or filter out noise you haven't been able to see before.
Something to ponder.
Definitely. That's all the time we have for this deep dive. Thanks for joining us.